Welcome back to the deep dive. Today, we are tackling what might be the most incredible journey in all of human biology, the 9 months of development.
It really is. We're going straight to the source material in human anatomy to understand, how we go from a single cell to a fully formed person.
Right, from something the size of a period at the end of a sentence to trillions of organized cells. Our goal, for you listening, is to make this whole chronological story feel clear and accessible.
And to do that, we should probably start with a few key definitions because it's not just about getting bigger.
Exactly. So when we say development, what are we actually talking about?
Well, development is the whole process, the gradual modification of all our anatomical structures from conception right up to maturity. And the engine driving that is differentiation.
And that's how a cell gets its job, so to speak.
That's it. It's how a generic stem cell becomes, say, a heart cell or a neuron. It's all about switching certain genes on or off.
Okay, and we break this whole prenatal journey into a few stages.
Yep. Prenatal is everything before birth, which is the field of embryology. Then you have postnatal, from birth to maturity. But that prenatal period is broken down into three smaller chunks.
The pre-embryonic, embryonic, and fetal periods.
Correct. Pre-embryonic is basically fertilization through getting implanted in the uterus. Then the embryonic period runs to the end of week 8. That's when all the major systems are laid down.
And the fetal period's everything else until birth.
Right. Mostly growth and refinement.
Okay, let's start at the absolute beginning then. Fertilization. I think we often forget just how logistically challenging this is.
Oh, it's an odyssey. For starters, it has to happen in a very specific place, the ampulla of the uterine tube, and in a very tight time window, usually within a day of ovulation.
And the journey for the sperm is just immense.
It's incredible. We're talking about navigating from the vagina all the way up the tract. It's a hostile environment by design, a filter.
A very, very effective filter. The numbers are shocking.
They really are. An ejaculate might have 200 million sperm, but you know how many actually make it to the ampulla? Fewer than 100.
Wow. Fewer than 100.
It's an insane attrition rate. And that's why, clinically, functional sterility is defined as a sperm count below 20 million per milliliter. If you don't start with overwhelming numbers, the odds of anyone finishing the race are basically zero.
And even for those 100 or so survivors, they're not quite ready to go.
No, not at all. They have to undergo a process called capacitation inside the female reproductive tract. It's sort of a final activation step. Only then can they even attempt to fertilize the oocyte.
And then they face the oocyte's defenses.
Its bodyguard, yeah. The corona radiata. The sperm's head has something called an acrosomal cap, and it releases an enzyme, hyaluronidase, to dissolve the cement holding those protective cells together.
And this is the really interesting part. It's not a one-sperm job.
Not at all. It takes dozens of them, all releasing that enzyme, just to clear a path for that one single sperm that gets to fuse with the oocyte. It's a total team effort.
So once that one sperm fuses, what stops a second one from getting in? Because that would be a disaster.
A complete disaster. That's called polyspermy, and it's non-viable. The moment that first sperm fuses, it triggers oocyte activation. The oocyte's membrane instantly changes, creating a block that prevents any other sperm from entering.
It slams the door shut.
Instantly. And that same trigger also tells the oocyte to finally complete meiosis, its final cell division.
Which sets up the grand finale. The fusion.
Right. The oocyte's nucleus becomes the female pronucleus, and the sperm's nucleus swells into the male pronucleus. They migrate to the center of the cell and fuse together.
And that process is called?
Amphimixis. That fusion, that moment, is fertilization. The new single cell, the zygote, now has its full set of 46 chromosomes.
So from there, we enter gestation. 9 months, 3 trimesters. The first trimester is, well, it's famously the most dangerous period.
It absolutely is. It's the critical period. In those first 12 weeks, the basic blueprints for every single major organ system are laid down. The complexity is just off the charts.
And the failure rate reflects that.
It does. Only about 40% of conceptions actually make it through the first trimester. A lot of that is due to chromosomal abnormalities. Our source says at least 10% of all zygotes have them. Nature has a very strict quality control process right at the start.
So what are the big steps during this high-stakes period?
There are 4 key processes happening: cleavage, implantation, placentation, and embryogenesis.
Okay, let's take them one by one. Cleavage.
Cleavage is just a series of rapid cell divisions. The zygote splits into smaller and smaller cells called blastomeres. It doesn't get bigger, it just divides, forming a solid little ball of cells called the morula.
And the morula becomes the blastocyst.
Yep, around day 5, it hollows out and becomes a blastocyst with a fluid-filled cavity. And it already has two distinct cell types. The outer layer, the trophoblast, which is all about nutrition,
And the inner cell mass.
which is the cluster of cells that will become the actual embryo. Those are the stem cells for the entire body.
Okay, so the blastocyst is ready. Next up is implantation.
Around day 7, it sticks to the wall of the uterus, the endometrium. The trophoblast cells on the outside form this aggressive, multinucleated layer called the syncytial trophoblast.
And it uses that same enzyme as a sperm, right? Hyaluronidase.
The very same. It uses it to literally erode its way into the uterine lining, creating little pools or lacunae that fill with the mother's blood. That's the first connection.
And once it's burrowed in, that inner cell mass gets to work on embryogenesis.
It flattens into a two-layered disc, and then, through a process called gastrulation, the cells migrate and reorganize themselves into the 3 primary germ layers.
And these 3 layers are the foundation for everything. Let's really lock these in for everyone listening.
Absolutely. Think of them as 3 master blueprints. First, the ectoderm. That's the outer stuff. It forms the surface of your skin, the epidermis, and this is huge, all of your neural tissue. Brain, spinal cord, everything.
Then you have the endoderm.
The endoderm is the inner lining. It forms the lining of your respiratory tract, most of your digestive tract, and glands like the pancreas and thyroid.
Which leaves the mesoderm doing pretty much everything else.
I mean, it's the powerhouse. The mesoderm builds all of your muscle, your skeleton, your heart and blood vessels, your kidneys, your gonads, all the structural and connective tissues in between.
And while all of this is happening, you have these support structures forming, the extraembryonic membranes.
4 of them, yeah. You have the yolk sac, which is a super important early site of blood cell formation. Then the amnion, which creates the protective, fluid-filled sac, the amniotic fluid.
Alontoa.
Its base eventually becomes the urinary bladder. And finally, the most important one for nutrition, the chorion.
And the chorion is what really builds the placenta.
Exactly. It's made of mesoderm and trophoblast, and it develops these finger-like projections called chorionic villi that extend into those maternal blood pools. This is the interface.
A key point here, though.
Mm.
The mother's blood and the fetal blood never actually mix.
Never. It's a critical barrier. Nutrients, oxygen, and waste all diffuse across the membranes of the villi. Blood flows out to the placenta through 2 umbilical arteries and comes back, loaded with oxygen, through a single umbilical vein.
The placenta is also basically an endocrine factory.
Oh, for sure. It's pumping out progesterone and estrogens, but also HCG, that's the hormone pregnancy test detect, which keeps the ovary producing hormones early on, and HPL, which gets the mammary glands ready for lactation.
Because that placental barrier is so intimate, it brings up the topic of teratogens.
A really important clinical note. Teratogens are anything that can cross that barrier and disrupt a development. Pesticides, certain drugs, alcohol.
And the source specifically calls out Fetal Alcohol Syndrome, FAS.
It does. It states it's the #1 cause of mental retardation in the U.S. Smoking is another major one. It reduces oxygen delivery to the fetus, plain and simple, which can lead to all sorts of complications.
Okay, so that's the whirlwind of the first trimester. The second trimester is a bit calmer.
Much calmer. It's more about growth and refinement. The organ systems are basically all there, and they're just maturing, getting close to being functional. The fetus starts to look much more, well, human.
And the third trimester is the final sprint.
It's all about rapid growth. This is where the fetus packs on the most weight, about 5.7 pounds on average. The organ systems become fully functional, which is why the chances of survival for a premature baby increase so dramatically during this time.
The uterus itself is also undergoing a massive transformation.
Oh, it's huge. It can grow to be 30 centimeters long, and the whole package, uterus, fetus, fluid, can weigh around 10 kilograms.
And all that stretching and pressure eventually leads to parturition labor.
Right. It's a hormonal cascade. Rising oxytocin levels make the uterus contract, starting from the top, the fundus, and pushing down.
Labor has 3 stages. The first one, dilation, is usually the longest.
Oh yeah, it can be 8 hours or more. The cervix is slowly dilating or opening, and contractions get progressively stronger and closer together. It usually ends when the amnion ruptures.
The water break.
That's the one.
So once the cervix is fully dilated, we're into the expulsion stage.
Right, this is the delivery of the baby. It's much shorter, usually under 2 hours. This is also when procedures like an episiotomy might be done to prevent tearing, or if necessary, a C-section.
Which our source says is pretty common.
About 15 to 25% of deliveries in the U.S., yeah.
And the final stage,
No.
It's not over once the baby is out.
No, you still have the placental stage. The uterus keeps contracting to push out the placenta, what's called the afterbirth. That usually happens within an hour.
We should also quickly mention breech births.
Right, where the baby is positioned feet or buttocks first. It only happens in 3 to 4% of deliveries, but it comes with risks, like the umbilical cord getting compressed. It's also often associated with other congenital issues.
And that takes us to the other side, the neonatal period, that first month. This is maybe the single most dramatic physiological transition a human ever makes.
I mean, think about it. You go from having everything done for you by the placenta to having to do it all yourself, instantly.
Starting with breathing.
That first breath is everything. It has to be this massive, powerful gasp to inflate lungs that were collapsed and filled with fluid just moments before.
And that single breath completely rewires the circulatory system.
It does. The pressure change from that breath slams shut 2 fetal bypasses. The foramen ovale, a hole between the atria of the heart, and the ductus arteriosus, a vessel that shunted blood away from the lungs, snap, snap. And just like that, you have the adult circulatory pattern.
And their vital signs reflect that massive effort.
Absolutely. A newborn's heart rate is way up there, 120 to 140 beats per minute. Their breathing rate is around 30 breaths per minute. They're working hard, and their kidneys can't concentrate urine yet, so they lose a lot of water and need a lot of fluid.
Okay, let's wrap up by connecting a few of these embryonic processes to the adult anatomical patterns they create. Let's start with the big one, the nervous system.
So it all starts with the ectoderm, the outer germ layer. A section of it thickens into the neural plate, which then folds up to form the neural groove, and the edges of that groove fuse together to create the neural tube.
And that tube is the future brain and spinal cord.
The entire central nervous system. But what's really cool are the cells right at the edges of that folding plate. They break off and form something called the neural crest.
And the neural crest cells are travelers.
They migrate all over the developing body and form the entire peripheral nervous system. All your sensory neurons, your ganglia, even parts of the adrenal gland.
Amazing. Okay, what about the skeleton? How does the spine form?
The spine forms from blocks of mesoderm called somites. A part of the somite called the sclerotome migrates in clusters around a central rod, the notochord. Those clusters become the vertebrae.
And the notochord mostly disappears.
Almost. A little remnant of it stays behind in the adult as the soft, jelly-like center of your intervertebral discs, the nucleus pulposus.
And knowing that helps you understand birth defects.
It explains them perfectly. If that neural tube doesn't close properly, the vertebrae can't form correctly over it, and you get spina bifida. Same with the face. If the different parts don't fuse right, you get a cleft lip or palate.
One last example. The reproductive system's in different stage.
Yeah. For the first 6 weeks, male and female embryos are anatomically identical. They both have 2 sets of ducts, the mesonephric and the paramesonephric ducts.
And testosterone is the switch.
Testosterone is the switch. If it's present around week 6, the mesonephric ducts develop into the male system. If it's absent, the default pathway takes over, and the paramesonephric ducts develop into the female system.
So this has been a massive journey. We've gone from the single-cell zygote through the risky first trimester where the germ layers set the stage. We saw the placenta as this vital lifeline, and we followed the process right through labor and the incredible transition the newborn makes.
And the really powerful idea here is how those embryonic patterns persist. The structures that arise from the neural crest or the somites, they maintain their relationships in the adult body. If you understand the blueprint, the final building makes so much more sense.
Which leaves us with a great question for you to think about.
Right. How does knowing the embryonic origin of a tissue, say, knowing that all muscle comes from the mesoderm, help you predict what other systems might be affected if there's a congenital defect in that one germ layer?
A fantastic way to think about how those first few weeks really do shape our entire lives. Thank you so much for joining us for this deep dive.
We'll see you next time.